Good morning. Welcome to Homecoming 2018. I'm Larry Jameson, Dean of the Perelman School of Medicine. We've got a wonderful event for you and glad to see all of you out early this morning after Penn's victory last night in the Palestra against Rice. And just behind us later on, Penn will play Harvard, and there'll be another victory against Harvard football. But today we're going to talk about victories against cancer. And you're going to hear about the ImmunoRevolution. Really exciting science, really exciting clinical trials and translational research. We've got a wonderful group of panelists who are truly at the cutting edge, and it's in many ways the best of Penn. And I'm so happy for all of the alumni to have a chance to come back and reconnect with your friends, with faculty that you might have worked with family, some of you, and with the institution. The University of Pennsylvania is a thriving institution, as you know, in the field of research. Reuters ranked us as the #4th most innovative university in the world. We've been climbing that ranking, and today you'll see just a few examples of how we've earned that. I also want to mention that the Power of Penn campaign is well underway. Our goal is to raise $4.1 billion as part of this campaign. We're well on the way, and I wanted to highlight 2 or 3 things that are important in the campaign at Penn Medicine. Now, if you're rediscovering the campus, you might have noticed a large construction site immediately across from the Hospital at the University of Pennsylvania, HUP, where Penn Tower used to be. That's our new patient pavilion. It's a magnificent clinical facility. It's been designed by one of the world-leading architects, Lord Foster. And in addition to its beauty, it's going to be incredibly functional for clinical care. It's being designed for the future, where doctors and nurses can provide patient-centered care with state-of-the-art facilities, and that will open in 2021, so not very far away. Another key objective is to support our medical students and graduate students, so support for education, financial aid. We're fortunate to get a really elite group of trainees here, and they make important contributions to our research, to our clinical environment, and they help keep our faculty on their toes, and you'll hear from some of the faculty later on. And the third element is to support the faculty with chaired professorships, and you'll see, just as I close, a video about the ImmunoRevolution. And I just want to end by pointing out the importance of philanthropy to support our creative research. You'll hear from one of the panelists today, Dr. Carl June, who 20 years ago received support from the Abramson family. They helped bring Dr. June to the University of Pennsylvania, and 20 years later, we're certainly reaping the benefits of that philanthropic gift. This research, both for the clinical trials and the discovery, is very expensive to carry out. The philanthropy we've received has been arguably the most important part of allowing the scientists to be creative and move more quickly. So welcome back for Homecoming and Alumni Weekend, and we can now play the video about the ImmunoRevolution.
When you think about a revolution, you think nothing will ever be the same again. And we feel that way with immunotherapy for cancer.
April 11th in 2012, and that's the day we treated Emily Whitehead, our first pediatric patient with CAR T cells.
She had a very high fever one night, and when it broke, she had no evidence of leukemia. Her T cells were working.
Now we have the hindsight, almost 5 years later, she's still in remission and she's a normal child.
What's exciting for me is to meet patients who've been treated and to see them go back to their everyday lives.
Every single patient I have seen go through this has gone through this with courage and dignity and really have led the way and provide hope to so many more people.
It's changed the way I practice surgery in the way that I'm much more hopeful, so it's really exciting.
So the therapy that we started here in Philadelphia was initially 3 patients from our local Delaware Valley catchment area, and now our trials are international. That's really an exciting event and just the beginning of globalization of this new kind of therapy.
We have seen a remarkable thing happen in cancer immunotherapy here at Penn. That 20 or so years of basic immunology discovery led to the tools that could make this kind of therapy today. So we can now build on these cancer discoveries and these cancer successes to use the immune system for many other diseases as well.
So we're all interested in waking up the immune system and using different elements of that immune system to develop therapies.
We have everything here. Really, I think it's the only place right now in the United States where everything from conception of the idea to delivery in humans can be done in just one institution.
There is a unique spirit and unique group of people here at Penn that really has allowed this to happen here, to happen now. The idea that this type of therapy can be developed in the course of 7 years from the first patient treated to FDA approval is astonishing.
Quite honestly, the FDA approval of CAR T cells means that yes, we can win this. We can do something that's never been done before.
The FDA approval really gives a mark that we've made something happen, and it happened here in Philadelphia at the university, which is an incredible landmark.
It's like landing on the moon, pretty much. You know it could happen, but you never thought it could happen. So there we are.
All the components are in place to be able to make these great breakthroughs. This goes way beyond cancer. What we need, frankly, are resources to do this faster and more efficiently.
Philanthropy has a role now to accelerate this so that it becomes universally available as rapidly as possible.
So here's our promise to you. We're going to move fast, we're going to be impactful, we're going to get this done, and we're going to improve the lives of patients.
Well, good morning, everybody. And thanks for being here this morning. And welcome to all our Penn alumni back to the university. And let's get this right from the beginning. Go Penn, beat Harvard. That's why we're here. My name is Bob Vonderheide. I'm the director of the Abramson Cancer Center. I've been at Penn for 17 years, and I've been the Cancer Center director for the last year and a half. And let me tell you what a year and a half it's been. I can tell you that the Abramson Cancer Center and the Perelman School of Medicine here at Penn are literally on fire. It's amazing what's going on. Today, I'll be the moderator of a discussion with a number of my colleagues. We'll take your questions after we have a chance to discuss a little bit about some of the exciting activities. You know, here at the Abramson Cancer Center, there is an upward trajectory that has caught national attention. Every metric is up. Research dollars, grants coming in, the number of patients we're taking care of, clinical trial accrual, and breakthrough discoveries are all increasing. This year alone, as you saw in the video, there have been a number of FDA approvals based on investigations led here at the Abramson Cancer Center. This involves, as you saw, not only children and young adults with leukemia, but now adults with non-Hodgkin's lymphoma, adults with breast cancer, adults with a rare neuroendocrine type of tumor. This has all happened in the year and a half based on science done here at the Cancer Center. In fact, national rankings now list us in the top 10 of all cancer centers in the country. This is 2 years in a row we've received that designation, the first time in the history of the Cancer Center, really reflecting the great upward trajectory. We are so pleased to be part of this great university. It's really a secret sauce. Part of the critical aspect of our success has been our expertise and our clinical translation in immune therapy. And that's what we're going to talk about today, the ImmunoRevolution. And this is where we use the immune system to attack cancer using drugs and other genetically engineered cellular therapies to create great results for our patients. And then to see and talk about today if we can go better, do better, and apply these to other types of diseases beyond cancer. So in Philadelphia, we're used to revolutions. It goes back hundreds of years. And we know one when we see one. And this is where we are. And we will talk about this today. What I'd like to introduce are the revolutionaries, who are our colleagues here today. And let me quickly tell you who's on stage, and then I'm going to go see what's on their mind this morning, and then a chance for you to ask them questions as well. So in the middle, Carl June. Carl June is the architect of the chimeric antigen receptor T-cell therapy that you heard about in the video. He's the director of Penn's Center for Cellular Immunotherapy, and recently named by Time magazine within the top 100 most influential people in the world. David Porter, on your far right, leads the cell therapy and transplant program at Penn and is a leading clinical investigator of the clinical trials that led to the CAR T-cell approval. John Wherry, to your left, is the director of the Institute of Immunology. He's newly named chair of Penn's Systems Pharmacology and Translational Therapeutics Department, and he's a world-class immunologist who's put the concept of immune exhaustion on the map. Noelle Frey, next to John, is our lead clinician treating patients with acute leukemia and chronic leukemia using CAR T-cell therapies. She's the associate director of Penn's bone marrow transplant and cellular therapeutics program. And Avery Posey, in between, next to Carl, it recently completed a postdoctoral fellowship in Carl June's laboratory, now runs his own laboratory and exploring the next generation of CAR T-cell therapy. No one does team science better than Penn, and we hope to have a chance to chat with you about that this morning. Let me start by asking Carl June the first question. Carl. Okay. So Penn became national and headline news when Kymriah became the first FDA-approved personalized gene therapy for cancer. And my question for you, Carl, is this is really a 20-year overnight sensation. How did we get here? How did this happen at Penn?
Well, it's not an accident, actually. So Penn has had a history of being within the School of Medicine and one of the best immunology programs in the world. So world-class graduate program in immunology. And then a strategic decision was made back in the mid-1990s to go into cell and gene therapy, and go to harvest some of the great basic science that we had. And so they recruited people to lead the cell and gene therapy programs. One was Gene Bennett, who has helped now develop a curative therapy for blindness. And I was recruited and came to Penn in 1999 to establish a program in cell therapy and gene therapy in cancer. And so with that, I met David Porter that first year, and we talked about how we could develop this. And it's complex. We wanted to take the patient's own T cells and then genetically modify them into killer T cells and give them back. And no one ever thought that could really be a therapy other than maybe some kind of experiment. And so we were kind of under the radar screen. And then, we began treating patients with various kinds of cells with leukemia in 2001 and, kept improving things. Then in 2010, we made, after a lot of mouse experiments and making better of these modified T cells, we first treated some patients David had that had chronic incurable leukemia, and we had amazing results. 2 out of 3 patients are alive today, 7 or 8 years later, and seem to be cured. And the other patient had a response as well. So that was published. They treated him 2010, 2011. It was on the front page of the New York Times and New England Journal of Medicine, and the firestorm took off. That showed that it could work, but then no one ever thought it could become actually a commercial product beyond some sort of boutique, and that's the amazing thing. And August 30th this year, the FDA approved it with a unanimous vote. and now it's the first commercially approved cell and gene therapy.
An amazing story of dedication and vision on your part, Carl. You know, as that drug was developed, a toxicity emerged, a new toxicity never been seen before, and we had to learn how to manage patients and get them through that. And actually, on the day the drug CAR T-cell was approved, the FDA approved another drug to treat the side effects. And Carl, how did we figure that out?
Well, we're blessed with having, really good laboratory investigations. We studied the patients and their cells. So what happens is the patients get very high fevers, but it's actually the result of their immune system killing the tumor. So David's found that if they don't get the fever, they basically those are the patients who don't respond. But the initial patients, we didn't know that. We thought they had infections, to be honest. But then on the third patient, we were measuring markers in the blood of what could cause fever, and we found that they all had, at the time of the massive fever, and I'm saying 106-degree fevers is what they get.
106.
106, and there's no infection. So what could that be? Well, it turned out that they had very high levels of a protein called interleukin-6. There was not thought to be any therapy for that in cancer to treat that, but my daughter has a form of arthritis, and that is treated with a drug called tocilizumab that blocks high levels of interleukin-6. And so Emily Whitehead had 106-degree fever and was, in 2012, near death. She'd been made do not resuscitate. We found her levels of interleukin-6 were 1,000 times above baseline. So when we got that, I called the physician over at Children's Hospital, and within hours they were able to get the drug tocilizumab because it was on the formulary there to treat rheumatoid, arthritis patients, and then it made a miraculous response. Within hours, she basically woke up. So the FDA did something they've never done. When they approved last year the Kymriah, which is our CAR T-cells, they co-approved the use of tocilizumab to treat the fevers. So we now have taken something that it would have been a showstopper, basically, because patients would have died. And now in the registration trial that Novartis did, not a single patient died from these high fevers.
I think it speaks so much to the team that you had built, that the treating physician sent a sample to the lab, they got a result hours later, confirmed it, called Carl. I think you were on the tarmac somewhere. Good thing your cell phone was still on. You called Steve Grupp. Steve Grupp spoke to the ICU nurses, and that little girl's life was saved in a matter of hours, had it not happened. It's just amazing. David. So David Porter, we're talking about a lot of extraordinary events. But this is becoming ordinary. This is now how we're treating certain types of cancer. How are we going to make extraordinary work like this become ordinary? Tell us about bringing this to the national stage and disseminating this around the world.
Yeah, well, good morning. Thank you. How do you make the extraordinary ordinary? How do you go from something that we treated truly 3 patients, that's all we had resources to treat, to now developing and disseminating it as a commercial product, yes, across the world, and also developing it beyond the current applications. It's been extremely complicated. It's extremely rewarding. At Penn, what we were really good at, we had a program where we could give cells, we could give human cells to treat cancer. That's what bone marrow transplant does, and we had done that for years. We have a very good infrastructure to do clinical trials, but nobody was expecting this kind of explosion. So what Carl described was something that I think we do better than anywhere that I have ever seen, and that is collaborate and work as a group. And just, looking at my friends and colleagues sitting here from every walk of this discipline,
we have been able to come together and do something unique and different. We have developed this just incredible expertise in a new field of medicine. Truly a field that had not existed 8 years ago. And so we have that expertise. We have been able to put a team together here at Penn to dramatically expand how we treat patients. That requires incredible resources, it requires incredible people. It attracts patients from all over the world, in fact. So we have developed systems to be able to take care of them clinically. We need space for them. We went to Dean Jamison and said we need a new hospital to house them, and the next day, a building started going up across the street that you'll see when you walk out.
We appreciate that very much, Larry.
I'm not sure it worked exactly in that timing, but we do. We need to grow and we need to expand to be able to manage this. But we also have an obligation to be able to disseminate this expertise across the country and across the world. So one of the things we're doing now is we are developing programs to provide that education to colleagues, as well as patients, but to colleagues so that they learn how they can manage these kind of unique therapies in a setting that it didn't grow up in, you know, in a completely new setting. And so that takes people and it takes dedication and time.
Well, it's a whole new medicine, and we're proud to be teaching the world how to get this done right. We've had a number of very important partners to get this done, including industry. And I wonder, David, if you could reflect a little bit about our partnership with industry and developing this new therapeutic.
Yeah. When we started, we had limited resources. We were able to treat 3 people, and it became very clear that this was very complex, very complicated, and frankly, tremendously expensive. And I think, hugely to Carl's credit, it became very clear we needed a partner. And it was an exciting but complicated process, and ultimately, Penn developed an alliance with Novartis. Often you hear about academic clinical trials funded by pharmaceutical companies. This wasn't Novartis funding us. This was an alliance. This was Penn and Novartis working together, and really it was a model that had never been done before. This was really unique. It was a collaboration. It allowed us to treat these patients and move this field forward at a pace that just wasn't going to be possible, I think, here at Penn alone, or that a pharmaceutical company could do by outsourcing it to a number of institutions without the kind of expertise that we had here. And I think that was tremendously synergistic, and it led to just such enhanced development of this therapy.
Yeah, in many ways, the partnership with Novartis was a new model for academics and industry to work together, and it's been a learning curve for sure, but we see now FDA approvals. The industry has become a $30 billion industry across the world, and we still feel Philadelphia as the home. I think Carl and his colleagues have discussed this notion that this city is Silicon Valley. And there's amazing activity. I wonder, Carl, if you could speak to Silicon Valley here in Philadelphia.
It is really one of the unanticipated benefits. So because of Penn's strategic vision to build cell and gene therapy back in the 90s, all the rest of the Ivy League schools missed that. This doesn't exist in Boston, and it doesn't exist in our competitors on the West Coast that I won't name. So, and what happened, back in the 1950s was that the first flu vaccine was developed. And to make vaccines, you had to grow eggs, and then the vaccine was made, the Sabin and Salk vaccines. So a lot of expertise in making vaccines ended up being an industry here. And there's many pharmaceutical companies around here, such as Wyeth and GSK and so on. And that's all because of that vaccine thing, 50 years ago. Now what we have is, and then, in the San Francisco Bay Area, the Silicon Valley happened because of some advancements in computer technology at Stanford, and then that led to a giant industry there with hundreds of companies and billions of dollars per year of new growth and jobs. That's happening here now. And we already have about at least 5 companies that have been started, hundreds of new jobs, and there are plans to build many plants and office buildings and so on here, so that this area will have, I think for decades and decades now, a legacy of new jobs and a new industry that will be headquartered here.
Yeah, it's absolutely amazing what's happening here in this city. I want to bring John Wherry into the conversation. John, as you remember, is a basic immunologist. None of this would have happened without basic science driving the insights. John, as you heard, is a new chair of our systems pharmacology. So the institution has asked John, as an immunologist, to lead pharmacology. John, how does that make sense in 2018?
Yeah, thanks, Bob. It's a fascinating question for a number of reasons. It's I think, a very bold move by Larry and the School of Medicine to think this way. But what it reflects is a bit of what you're hearing. The pioneering work by everyone else on the stage in figuring out how to engineer immune cells and turn them into drugs really speaks to a larger mission and a larger set of opportunities where the immune system is now becoming one of the major toolkits in our ability to treat diseases. And this is different from the traditional way we think about drugs. We usually think about a drug, you take a drug and it targets the diseased cell or the symptom that's causing your disease. Well, what you've just heard about is something completely different. It's taking the immune system, in this case, engineering it outside the body, and training the immune system to become the drug. And we're now treating thousands of patients with these kinds of engineered immune cells, and there are hundreds of thousands of more patients being treated with drugs targeting the immune system and effectively retraining the immune system to become the drug. This is a new concept in pharmacology and how you think about drug development. And it leads to a new concept in medicine in general. If we can retrain a system of the body like the immune system to become the drugs, we have to think differently about what pharmacology really is. And that's a huge opportunity and a really bold vision for the School of Medicine.
So do you think this new drug, which we need to understand the pharmacology, cell therapy, do you think it has relevance for other diseases, HIV, multiple sclerosis, autoimmune disease? How do you see this coming forward?
Well, I think this is the big opportunity, and I think this is where Penn stands out really differently from other institutions. And that is the vision. What we're learning from what Carl and others described can now be applied for many, many diseases, infectious disease, autoimmune diseases, but even thinking creatively way beyond that, cardiovascular disease, neurological diseases. There are tremendous opportunities to employ the power of the immune system. And you can think about what Carl really pioneered in taking cells from the immune system apart and re-engineering them. You can now think about doing that in many other settings for many other diseases.
Yeah, we've talked about using these T cells to destroy the autoimmune cells, or using T cells to destroy atherosclerosis. In one of our brainstorming sessions, someone said, let's use the T cells to destroy fat cells. So, we could address a lot of things, I suppose. But it comes down to the patients. Our inspiration is our patients. Noelle is taking care of these patients on a daily basis and has really championed clinical care here. Noelle, you're a specialist in leukemia, and when you think about how you had the tools you had to treat patients with leukemia even 5 years ago, compared to the patient you saw yesterday, describe that difference for us.
Yeah, well, it's night and day. So I think first and foremost is just the very clear fact that we have more effective strategies now than we had even a few years ago. And you said patient I saw yesterday, but a patient I saw earlier this week with my medical student is a young woman who came down from New York in her early 40s. I had to explain to the medical student that being in your early 40s means you're young. So I feel more and more strongly about that. But we met this woman, and she was at a point in her care where she was running out of hope. Her leukemia had been treated with several different back-to-back therapies, and nothing had been able to get it under control. And in that initial office visit I had with her, I was able to offer her 3 different potentially very effective strategies. One was with a targeted drug that had just been FDA approved within the last year. And excitingly, the other 2 strategies were actually clinical trials. And I said to the medical student, when iPhone 6 was out, again, I'm trying to relate to my medical students, when iPhone 6 was out, we couldn't have offered any of these therapies, and that conversation would have been dramatically different and very disheartening. And I think in my clinical practice, it's great that I have this change of actually new therapies where the former extraordinary is becoming ordinary. But the other major change that I've seen as somebody who helps patients get through clinical trials with new drugs is just the breadth and the diversity of clinical trials that are available now compared to even 5 years ago. And I think we see this benefit that we saw in the video of individual patients benefiting from Kymriah. And I think one of the big downstream effects of that is, like, we know we can do it now. We know we can, and all these creative minds in the lab and in the clinic are really trying to take that to the next level. And it's really inspired just all these new clinical trial opportunities.
Absolutely. The inspired confidence you feel in the clinic and the energized conversations we have with our patients is amazing. I think, Noelle, and you can speak to this well, and Carl, you know, we still have a lot of work to do. These therapies, although fantastic, are not working in some patients, and in certain types of cancers, they're not working at all yet. We have a lot of work to do. So as we celebrate what's happened, there's a bittersweet quality knowing that we have to move better. But as you said, we now have a window on success. What are some of the ideas to improve these therapies further that you're most excited about, Noelle?
Well, so just that this particular patient still on that same track. So she has a type of leukemia where CAR T cells actually that's been developed already, wouldn't be able to target. And so one of the clinical trials that we have open now is another engineered CAR T cell that can target a different protein that happens to be on this patient's leukemia. And I think that's a very exciting area in oncology in general, and as we were just alluding to, other aspects of illness, where using the same technology to go after different targets, just retraining the immune system. And I think the other exciting things are, we're so excited about singular successes. What if we combine them?
Yes.
What if we combine different strategies together to get even better outcomes?
It's interesting, as you say, the gene therapy base for the CAR T cells is able to be adapted for different targets. We don't have to go back and design something completely new. We have a base platform now. Carl, I was wondering, as we were talking, do you think this will be relevant for patients who have solid tumors like prostate cancer or glioblastoma multiforme? Are you confident that we're going to get there?
Yes, Bob. I think we have the principle proven that this can work and be a general solution for cancer. The issue is how long it will take. In the U.S., approximately 600,000 people die every year from cancer, and about 50,000 of those are blood cancers that Noelle takes care of. And I think all of those will be, subject to having what we have in the near horizon, different kinds of cell therapies within the next 5 years. But the unknown is how long, the big unmet need in solid cancers are things like pancreatic cancer, brain cancer, prostate, ovarian, et cetera. And how long will that take? We don't know, but that's what we're focused on. We need to be nimble on that. We have some interesting trials underway here now in almost any cancer you can name, but they're early trials at this point. And so I'm very excited at the prospects. I want to make sure we manage our expectations. We don't know how long it will take.
Yes. Yes, absolutely. The cell manufacturing facility we have here just across the street is entirely devoted to new therapies, experimental therapies. The commercial manufacturing has now moved to the commercial entities in New Jersey, not part of Novartis, not part of us. Avery, I want to ask you, as you today represent the next generation of revolutionaries. I don't know if you're up in the sketch somewhere. Thinking about what Carl said, what are you excited about in immune therapies, and what do you see as the next frontier?
I think solid tumors is definitely part of the next frontier. At this point, CAR T cells in leukemia seem to be doing most, if not all, of the work. And for solid tumors, I think we need to have CAR T cells do some of the work, but make the rest of the immune system do the bulk of it. And so that's what I'm really interested in, is where can these engineered cells make an impact into the rest of our immune system to have a significant impact against the tumor. And so we're interested in CAR T cell trials that will go forward in breast cancer and prostate cancer and ovarian cancer. And we have a special target that we're interested in that will hopefully initiate next year with a CAR that we've developed in my group.
So tell us about that innovation, right? You found a new target. How'd you find it?
So actually, it really depends on work that others have done way before us. And adapting that to this modular platform of CAR T cells. The targets that we're interested in have been known for quite some time, but we haven't been able to use the immune system to target them. And CAR T cells, for the first time, give us the ability to do that. And now we need to figure out how do we add to that? What can we do in addition to the CAR T cells, either in combination or through modification of the T cell itself to improve those therapies? There's still a lot of other things to learn, including what causes some of the associated toxicities with CAR T cells, some of the transient neurotoxicities that are observed, and how can we modify the T cells or modify the CAR so that those occur less. In addition, we have preclinically, we have from labs at Penn and elsewhere, a wealth of new modifications to CAR T cells. Actually, when Penn and Novartis started their alliance, it was the first company in the field. Now the number, I think, is over 150 companies worldwide trying to duplicate the success that has been done here at Penn, either with CD19 CAR T cells or in new formats, and dozens of investigators as well. So there's a wealth of new ideas that have worked preclinically in our rodent models, and we know that many of those, if taken forward to human trials, will fail. And so we need additional models that are more relevant to the human system. And so actually we've collaborated with Penn Vet uniquely because we have a veterinary school on campus at Penn that's associated with our medical school, a short distance away, and we've collaborated to develop a large animal clinical model of CAR T cells. So we have dogs with spontaneous tumors, lymphomas and osteosarcomas that come into Penn Vet. We can develop CAR T cells for the dog's tumor and treat the dog, send them home, and that allows us to model CAR T cell activity in animals. It allows us to treat our animals, but then we can learn a lot from that and hopefully send some of these ideas that have worked very well in our rodent models through dogs, and then decide which ones actually are valuable and should go forward into humans.
It's a really important point you're making. We're going to explore it in a minute on another video, but you know, we are here based at this great university where we look around us and we see all these world-class other schools besides the Perelman School of Medicine. The vet school, you know, really top vet school in the country, nursing school, #1. We mentioned Children's Hospital, #1. This collaboration with the vet school is an amazing and a special part of how we can have Penn pride in this work, and I'm glad that you brought it out. The patients we're talking about are not, these are people's pets. And it turns out that osteosarcoma, which is a terrible problem in humans, and it's a rare but terrible problem in humans, is very common in dogs. So we can treat many more patients with osteosarcoma who are canines, treat them, they are patients after all, and bring that same therapy over to humans in a much faster way. I was going to ask Avery one more question before we show a video about that experience with the vet school and beyond. You're sitting next to this guy, Carl June. You were his postdoc for a couple years. What was that like? Because a lot of people out there want that job.
Yeah, I think it was a very unique experience. One thing that I think is different about Carl is that given his success, you would expect a different personality. Carl's a very genuine person that tears up every time he discusses a patient. And that is something that will stick with me for the rest of my days, is that this is the reason why we're actually here.
Well said. Thank you.
Thank you.
So as promised, let's take a look at this video which outlines this special relationship with the vet school as an example of the power of Penn, which is why we're all here today and this weekend. And after the video, we'll take some questions. Okay?
My dog is the sweetest dog I think I've ever met.
I adopted her while I was taking classes in medical school. She was a huge stress relief for me.
Dante is a great dog. Dante has, unfortunately, a aggressive cancer.
Dogs develop spontaneous cancers just like us, so dogs are a good model for human disease.
By collaborating, we are discovering novel ways to treat cancer in both humans and in animals.
We want to advance our knowledge, and we're using our best friends to do that.
Research becomes impact when it actually changes people's lives. And I think it's one of the key strengths of the University of Pennsylvania.
I came to Penn to work with Carl June. Cancer immunotherapy is our next generation and best generation of cancer therapies.
With this UPenn technology, we have dogs that were diagnosed with a terminal cancer in 2012 still coming back to see us. And that's just unbelievable.
We can treat the dog, but then we can learn something from that treatment that we can apply to kids and adults with cancer. This work is definitely a moonshot in our field. It's a breakthrough.
This treatment is going to change the paradigm of medicine as we know it.
This type of therapy has just received FDA approval to treat cancer in humans.
That's the day you call mom and you say, hey, I did something today. It's an impact that now goes worldwide.
To be trained during this time is it's surreal. It's surreal.
We couldn't do all the good that Penn seeks to do without the engagement of the Penn family. Alumni, parents, friends, everyone who supports Penn are essential to the university's world-changing work.
When someone provides financial support to Penn, the real payback comes from seeing what your dollars can do.
I think it is possible that we will cure cancer, and Penn will lead the way in that.
Yes? Is it going? There we go. Great, great. So just a word about, you saw that celebration when Carl was standing on a coffee. That happened last August, the day the FDA approved the drug. And we sent an email out saying, everybody come. We called it the flash mob. And everybody come down. And I thought maybe 100 people would come, but 4,000 people came. We gave them T-shirts, so that helped. And you see the T-shirts on some of the folks today. So let's take a few questions. Really appreciate your interest. And we have folks with microphones here. If you don't mind, tell us who you are.
My name is Jules.
Jules, how are you?
Okay. Well, it's, the question is really a type of question. You know, the first thought was, why doesn't the immune system normally do this?
Yes.
Well, I have a melanoma, or had one, okay? And I discovered that the immune system does do certain things, like attack melanomas and change them in a way that they cannot replicate again, okay? That's like a little bit like the town that I live in. They have so many deer, they want to get rid of them. And they said, well, why don't we just kill all of them? And somebody came up with the idea, well, just sterilize them. This doesn't mean that the deer is dead, but it means that it can't procreate. So is it possible also to use this therapy rather than to kill, but to change it in a way that can also alter its ability to procreate or replicate?
I think you asked the most important question that is facing scientists. It's a really tough question, which is why I'm going to ask John Wherry to answer.
Yeah, it's a perfect question. So the immune system does normally recognize many tumors. And if not for the immune system, those tumors would grow faster. And the immune system kind of keeps things in check. And the job of the immune system is to balance that ability to deal with all the germs and invaders that might come from the outside and fight them off really potently, but then not damage your own tissues. And cancer fits into this really difficult place for the immune system, right in between. It looks a little bit different than our normal tissues, but then a little bit the same. So what happens often is that the immune system tries, but over years, it literally gets tired and becomes something that we in the field have called exhausted. And especially in melanoma and lung cancer and kidney cancer, one of our jobs now is to revitalize, is to wake that immune response up. And the goal there really is to kill the tumor cells, prevent them from replicating, but also get rid of them. Don't leave a chance to have some of them sit around and wake up later. And the amazing thing about biology is that if you give it time, it will figure out a way around some of the blocks you put in place. So the goal is to wake up those tired immune cells that do recognize the tumors, or if you don't have them recognizing the tumors, to engineer your immune system to do so and prevent that exhaustion and that tiredness that we see in a lot of cases.
Well, how do these cells wake up later? The cells that have been injured, let's say, by by the body's immune system. You said they sit around and then they wake up later.
So our goal is to get rid of all of them as quickly as we can. Don't leave enough around. And that's the goal, is to really get complete eradication quickly.
Let's question over here. Tell us again who you are, maybe what school you're associated with.
Well, thank you. Good morning. I'm Scott. I'm Wharton 88. And thanks to Penn Medicine, I'm actually in remission of Ewing sarcoma. So thank you. And thank you guys. You give a lot of people a lot of hope. I have two questions. One, how do you prioritize the targets that you're working on? If you have a rare cancer, a sarcoma, a childhood cancer, are we last in line? Is it work the big ones? And so maybe you can talk about that. And just quickly, the second question, I love how you're working with the vet school and you're mashing teams up and you're being creative. I'm involved with software and systems, and I'm wondering, do you ever mash up with systems engineers, software engineers, big data people? Because there's a lot of commonality in the code and the thinking and the organizing. Thank you.
So great. How do we find targets, and what about data science? And we can talk about both separately. So, Carl, do you want to give your thoughts about how we pick targets?
It's a very complex question on, the medical terms they use are unmet medical need. And you can define that in several ways. Almost everyone who gets pancreatic cancer, if it's, unless it's found by accident, they basically, if you look 5 years later, they've all died. And that hasn't really changed. I mean, there's a huge unmet medical need there. And the same thing in sarcomas, which occur in children and adults. And if you actually look at So sarcoma is a rare tumor, as you know, compared to, say, pancreatic cancer. But if you look at actually total lives lost, years lost of life, sarcoma is much overrepresented because many of the children that get it are young and then they're deprived of an 80-year life. So I was on a committee with the National Cancer Institute to look at this exactly question of how to prioritize. So sarcoma is basically, as you implied, it's ignored by most of the pharmaceutical companies, because it's not a big market. There's many different kinds of sarcomas. And so unfortunately, they're not doing research in it. And the NIH looked at their portfolio of grants that are funded in sarcoma. And when you actually graph cancers out by years of life lost, sarcoma moves way up higher than it is in its just incidence, which is rare. So we have programs in sarcoma here for that very reason, and then we if we can get a therapy that works, then it's how can we get a company interested in it, or would we actually just market that through Penn Medicine? And we've actually had those discussions.
So we have a lot of interactions on the second topic around data science and the power of biomedical informatics in getting our work done. Actually, as part of Dean Jamison's last strategic plan, very forward-thinking, we launched the Institute for Biomedical Informatics, led by Jason Moore, who we recruited here to Penn, and it's just taken off, and we're finding that those conversations are some of the most productive to be able to learn from big data. A lot of traps. It's harder than it sounds, as you well know. We're at the leading edge of that. We're really pleased. We're starting to use algorithms of patients' tumors to design vaccines that very specifically activate the immune system for that patient. We have clinical trials that use that technology underway right now. Can we take another question? Oh, here we go, right here. Sorry.
Hi, I'm Ira Sager, class of 1980, engineering school.
Great.
Proud parent of Monique, who's in the med school starting this year. So my question regards the viability of this for the masses. Okay, so it's a personalized medicine, very expensive to do. There's some companies like Selectus, I'm sure you're aware of them, pursuing allogenic, which is trying to get rid of the personalized features of the medicine so that you can make a big vat of this and give it to everybody. My question is whether Penn is working on this, and if so, how well do you think this might work relative to an actual personalized medicine?
Super quick answer is big time, and I'm going to ask David and maybe Carl to jump in on some of these concepts.
Of course. Yes, Penn is working on that, exactly. And in fact, again, there's a repetitive theme here. Carl's lab is a leader, in fact, in developing allogeneic cell therapy. So just to remind everybody that the therapy we're talking about, this CAR cells, as you're describing, we take somebody's own immune cells out. They go to a laboratory, they're genetically modified, and then they're brought back to us to infuse into the patient. That whole process takes about 3 to 4 weeks, and in some cases, maybe even 5 or 6. That's a long time when someone has cancer and it's growing. So what you're talking about is having third-party or off-the-shelf cells, taking cells from a healthy donor, making them ahead of time in big vats, having 100 samples in your freezer, and you can treat somebody tomorrow. It has different advantages and disadvantages. There are certain limitations with that kind of therapy, and there are additional risks. I am tremendously optimistic, just as the CAR T-cell therapy, as a personalized therapy has been developed, that we will overcome those limitations. I think the big advantage is that it is readily available, and with some really, really innovative and novel manipulations, we are going to be able to make those cells safe and I think practical. It's not there yet. The clinical trials are just getting underway in people. I think we hope to have our first trial with the off-the-shelf engineered CAR T cells probably by early 2019, right, or before the spring. And we will be able to treat people potentially much faster, being able to treat more people, potentially in a safer, more effective way. So it's a really great thought.
Did you want to add anything, Carl?
Yeah, I'd just like to say about the cost of this. So right now, cancer is, it used to be patients died so rapidly that the actual cost of cancer was nowhere near the cost at a national level as, for instance, treating diabetes, where it's not curative, but that people live a long time. And now the cost of cancer is going up because the therapies are getting more effective. What we actually need is a cure. And then that can be much more, even if it's expensive, it's cheaper than what we do right now, which is recursive therapies that are expensive. As an example, myeloma is one of the most common blood cancers, and it's 2% of all cancers in the United States, but it's 7% of all cost. So it's much more, there's a so-called financial toxicity there. What we need is a curative therapy. We, with the Wharton School recently, had a meeting on this called The Cost of a Cure, and to look at financially how to do this. So it's a very important issue. I mean, what we need is curative therapies.
It was
and then learn how to make them cheaper.
I'm sorry.
Yeah.
This conference that Carl is talking about held just a couple of blocks from here. People came in from all over the country to try to think about this question, how will we pay for the cost? And I thought it was amazing that at the same university where this whole thing was invented, and we're still trying to innovate, that we're having a conversation about how we're going to pay for it. And people asked me and said, is that a conflict for you? And I said, no, it's the same conversation. It's the type of conversation a university like this has. The FDA commissioner came and was the keynote speaker. That's how important a conference it was. Let's take another couple of questions. Yes, right? Yes, you with the, oh, we have two. Let's do both of you and then.
Hi, Robert Sandler, Wharton 52. I applaud the work that you guys have done. It's absolutely miracle medicine as far as I'm concerned. I'm a survivor of the TAVR, which you're all familiar with, which I had in February, and I'm here, and it's exciting. I wanted to mention multiple sclerosis. I've been active in that campaign for the last 20 years. What kind of progress is made in that area?
So multiple sclerosis is an autoimmune disease that we're all familiar with. We are doubling down on our efforts at Penn Medicine to attack that disease. Underlying that disease is the immune system gone wrong. We recruited Amit Bar-Or, one of the world's leader in immunotherapy for multiple sclerosis just a few months ago. He's already published a paper in the New England Journal with a new therapy based on that. I think it's the tip of the iceberg of what can be accomplished in autoimmunity, even using CAR T cells against autoimmunity. Either any of you want to, John, do you want to say a couple words?
Yeah, I mean, it's a great question, and it's a huge area of opportunity. Bob mentioned the recruitment of Amit Bar-Or, which was a collaboration between the Department of Neurology, the immunology community, and a bunch of other communities around campus, and represents, I think, what you've heard about a number of times from this group, which is really this sort of team-oriented approach to problem solving in medicine and in science. And the idea that someone like Amit can end up on campus in a Department of Neurology as a major member of the Institute for Immunology and the immunology community, and working across Penn and CHOP to acquire samples, think about new treatments and interventions in multiple sclerosis really represents kind of the best features of the way the Penn environment attacks major problems like MS.
Cancer has allowed us to understand how the power of the immune system and how to use it to treat disease. We're looking at the term immune health across all diseases. I think we want to be the university that puts forth immune health. So we have.
If I could just chime in that there is good evidence, at least in a select group of patients, that bone marrow transplant can be effective for multiple sclerosis.
Thank you for pointing
And our bone marrow transplant group is starting to work with Amit Bar-Or as well, sort of integrating neurology and non-cancer medicine now with bone marrow transplant in our cancer group. So it really is all
wonderful. Okay, a question. Yes, is it? Did he have a question? Thanks.
Hi, Greg Kasoy, current applicant. I'm actually doing similar research with CAR T-cell therapy, and I was wondering what you thought the biggest obstacle was.
You're an applicant to?
To the College of Arts and Sciences.
Excellent. So what do you think, you asked, what is the biggest obstacle? Yeah. Avery, you want to?
I think CAR T-cells have been successful in B-cell malignancies. And we see some clinical success in multiple myeloma, and I think there are other hematopoietic malignancies that we will see some success with some tweaking of the therapy, for instance, in AML. The current limitation is how do we get into solid tumors, and some of that is getting around some of the microenvironment that the tumor has that suppresses T-cell activity.
Noelle, from your point of view, what do you think is we have to overcome?
Yeah, I think for me, it's almost like I've had, interacting with my patients, I've had this taste of this potential. And there's still, you know, every individual patient that's suffering with cancer without a good treatment option, it's an unmet need for that patient. And so, so I think I'm so excited that these types of investigations to find, you know, you have to find more targets to treat more tumors, to treat patients who, you know, don't have good options. You need the funding to support these trials, to combine it with other therapies. You know, you come at the cancer with an immunologic approach, you come at it with a small molecule approach. We still have so much work to do, so.
Maybe time for one more question. Well, okay. You had that microphone, go ahead, in the red jacket. Great, thank you.
Yes, hi, Glenn Berger, class of '84, college. I'm a practicing physician and practiced 26 years. And first I want to say hats off to everybody for this incredible breakthrough in cancer. I've always felt that at some point in my life, it would be nice to see chemotherapy be a thing of the past. One thing I was wondering about is, for example, the young woman who was the first one treated with leukemia with CAR T, and I understand that something like 5 years out, they were still able to find the CAR T cells in circulation, surveying her body and, you know, preventing cancer from coming back. What actually happens in the body to allow these cells to perpetuate? Because normally cells would have a certain lifespan, and that would be it.
The persistence was the key feature of this drug. Carl, do you want to comment?
Sure. I mean, the immune system is known to have memory, and the memory is basically done on the basis of cells that can live a long time, either by themselves or their daughter progeny. So we know that a vaccine, for instance, against smallpox, a single vaccine, if it's a good vaccine, can last more than 50 years. And that's been shown by some lovely studies. And the way that happens is that the cells have persisted 50 years in the patients since they had that vaccine. And so Emily Whitehead, as you're right, our first leukemia patient, is now, she was treated in 2012, she's 6 years out, and she still has CAR T cells. The first patient that David Porter ever treated, and this was in 2010, it was August 1st, 2010, just came back 8.25 years after that infusion, and he still has 1% of all his T cells are CAR T cells. Amazing. So this is the first living drug, and it takes advantage of the immune system to make memory.
So one final question right here, ma'am.
Thank you. Actually, it's not a question, but it's a statement of gratitude for your intelligence, innovation, courage, and for giving people like me hope. In 1989, at age 2 years, my daughter was diagnosed with ALL. She was at CHOP. We were devastated. I called my advisor from med school, who was the chief of staff at Boston Children's. He said, don't come here, stay at CHOP. You've got Beverly Lang, you've got CHOP, you've got Penn. We stayed at CHOP, and she's 31 years old now and doing great. And I think what you're doing for everybody would have changed our lives so much. And people who've had cancer and had family members know how it destroys families and changes the trajectory of life. And I just want to thank you all so much. I can't thank you enough, and I really appreciate everything. And I'm grateful to do what I do, but what you do is incredible.
Well, we thank you for sharing that. I want to close by, thank you for those wonderful words and your confidence. And we'd like to say back to you all, thanks to you. This is happening because we're a community. Our patients are our dear friends. We treat them like family. And the rest of the Penn community has supported Penn Medicine and the Abramson Cancer Center in unbelievable ways. Almost every good idea that we've talked about today can trace back to a supporter and a scientist talking where the supporter says, let's get started today. That's a conversation Carl had with Leonard and Madeline Abramson and Barbara Netter, and on and on. The power of philanthropy is amazing. The power of this community is amazing. No wonder we're on the leading edge. I want to thank everyone for coming today, and let's go beat Harvard bad.